Hi, everyone. These are the lecture notes for Chapter 5, The Structure and Function of Plasma Membranes. Plasma membrane functions include defining the outer border of all cells and organelles. In other words, the plasma membrane is the structure that surrounds the organelles and the cytoplasm and holds everything inside of the cell. It's almost like, I guess you could say, the skin of the cell. Managing what enters and exits the cell. So anything that's going to come into the cell, such as nutrients, or anything that's going to leave the cell, such as some cells secrete products like hormones. And and as well as oxygen, carbon dioxide, ions, which are charged atoms. We tend to call them electrolytes. All of these materials enter and leave the cell, and they have to do that through the plasma membrane. Receiving external signals and initiating cellular responses. So if the cell is going to respond, it normally is going to respond to a chemical or a voltage change, and there are proteins in the plasma membrane that are responsible for those changes or for initiating those changes. And cells do not exist by themselves. They don't exist on their own in multicellular organisms like humans and plants. So similar cells form tissues. So they have to adhere to neighboring cells in order to form tissues. The fluid mosaic model of the plasma membrane was developed in 1972. I won't tell you why that year is important to me because, well, I just won't, but you can guess. But it was developed by S.J. Singer and G.L. Nicholson. These are the scientists that developed this model of the plasma membrane structure. And the word fluid means, in the illustration, it looks like all of these molecules are just solid, stuck in place. But if you can picture all of those phosphate heads as marbles, and if you had marbles in an open space like a box, you could sort of move them around and the marbles would move past each other. They would kind of slip and slide and move around past each other. So all of these molecules are able to do that. It's not like, say, this protein, for example. This is a protein, it's a channel protein that allows materials to enter and leave the plasma membrane. All of the blue structures are proteins. Well, all of the blue, not this one. This is a carbohydrate chain. But the rest of the blue structures are proteins. And these proteins don't just jump around. I mean, this protein couldn't just jump way over here, but it could slip and slide through these phospholipids. So that is what fluid means, the fluid part of fluid mosaic model. And mosaic means that the components are scattered. They're a mosaic of components. The components are phospholipids, cholesterol, proteins, and carbohydrates. And they give the membrane a fluid character. Now, it's not as fluid as water, but it is not a solid either. So the plasma membrane is a fluid structure. It's just more of a thicker fluid. The main component of the plasma membrane is a phospholipid, which we did discuss in Chapter 4, has a hydrophilic head. That is the phosphate head, which is really a phosphate group, combined with a glycerol. And a glycerol is one of the components of a lipid. So the phosphate head has a phosphate group, and this phosphate group makes the phosphate head hydrophilic. And hydrophilic means that it is polar, and that means that it is soluble in water. So that is the reason the hydrophilic heads face outward and they face outside the cell and inside the cell, because the fluid on the outside and inside is water-based. Then the hydrophobic tails are fatty acids, and they can be saturated or unsaturated fatty acids. Just to remind you, if it's saturated, then it's a straight chain of fatty acids, and it doesn't contain any double bonds between the carbons in the hydrocarbon chain. But if it's unsaturated, it contains at least one double bond, and that will typically make the unsaturated fatty acid bent, at least at one spot. But the most important thing about the hydrophobic tails is that they face into each other. They face each other. In the phospholipid bilayer, the hydrophobic tails face each other like you see here. The heads are on the outside, exposed to the watery environment inside and outside the cell, and then the tails face each other. Because they're hydrophobic, they're nonpolar, they're insoluble in water. These illustrations show you what would happen if you just took a large sample of phospholipids and placed them into, say, a beaker of water. They would naturally orient themselves this way. The hydrophobic tails would turn inward and face each other, and the heads would face outward, and they would naturally form a lipid bilayer sphere like this. So this is just something that happens whenever phospholipids are present in water. The protein component of the phospholipid of the plasma membrane are these blue structures, some of which you find on one side of the cell. This one, for example, is on the inside of the cell. And if they're found on either the inside or the outside, they're called peripheral membrane proteins. Peripheral because they're on the periphery. If they are found extending all the way through the phospholipid bilayer, then they are called integral membrane proteins. And these membrane proteins have various functions. Some function as transporters. They transport materials across the plasma membrane. Some function as receptors. Chemicals can bind to them, and then those chemicals will cause a change inside the cell. Some function as enzymes, which speed up or catalyze chemical reactions. And some function in binding and adhesion. Integral proteins have one or more regions that are hydrophobic, meaning that they're insoluble in water, and other regions that are hydrophilic, meaning they're soluble in water. And this is because in order to exist throughout the phospholipid bilayer, the part in the middle here must be insoluble in water and soluble in lipids. So this region must be hydrophobic. And then the regions here that are facing the watery environment and that are located alongside the hydrophilic heads, the phosphate heads, these regions either need to be polar or they need to be charged. They need to have a charge. So the region in the center needs to be nonpolar, and the region of the protein facing the inside and outside of the cell needs to be either polar or it needs to have a charge. So if we look at the amino acids, they're grouped based on whether or not they're polar, nonpolar, or whether they have a charge. So there are two groups that are nonpolar. There's the nonpolar aliphatic amino acids, and then there's the nonpolar aromatic amino acids. Remember, if they're aromatic, they have a ring structure. So these amino acids would be the amino acids that you would find on the inside where the hydrophobic tails are in the phospholipid bilayer. And then polar, negatively charged, and positively charged amino acids are going to be found on the outside or the very inside where you have the phosphate heads, because these are soluble in water. And then you have carbohydrates, and these carbohydrates take the form of carbohydrate chains, and they can either be attached to a protein or to one of the phospholipids. So if they're attached to a protein, they're called glycoproteins. And if they're attached to a lipid, they're called glycolipids. So the glyco portion of that word is what represents the carbohydrate. And they have different functions. One of the functions is cell-to-cell recognition. These carbohydrates are very important when doctors are determining tissue compatibility for an organ transplant. They want to make sure the donor and the recipient have compatible carbohydrates on the surface of their cells. Tissue compatibility is very important. Finding a match is very important. So these carbohydrate chains are used for that. And they're also used for attachment because since they're carbohydrates, they tend to be sticky. If you think about it, sugar is a carbohydrate and sugar is sticky. And then we have proteins called receptor proteins. And these receptor proteins bind with a chemical, and then that chemical causes some kind of change in the cell. Our immune system's T cells have a CD4 receptor. It's just called CD4, that's just the name of it. It is a glycoprotein that recognizes HIV as self, which is not good, not a good thing. This is the way that HIV is able to shut down our immune system because T cells are extremely important in immunity, in developing immunity. So HIV tricks our T cells into recognizing it as a self molecule instead of as a pathogen. And it's able to trick our cells into becoming infected. And if you can shut down the T cells, you effectively will be able to shut down the immune system, the entire immune system. Now, we mentioned that the plasma membrane is fluid. So the fluidity needs to be flexible, but not so fluid that it can't maintain its structure. So it needs to be a more concentrated or a thicker fluid. It doesn't need to be as fluid as, say, water. The fluidity is affected by the types of phospholipids. In other words, whether or not they contain more saturated fatty acid tails or more unsaturated fatty acid tails. The more saturated fatty acid tails, the more rigid the plasma membrane is. Because remember, saturated fatty acids come from animal sources and they tend to be solid at room temperature. The temperature will affect the fluidity of the plasma membranes. If the temperature is cold, the membrane tends to be more rigid. And cholesterol, which is located within the fatty acid hydrophobic layer, the fatty acid tails, is it will keep membranes fluid when the temperature is cold. and it will keep them from not getting too fluid when the temperature